our next speaker is going to be Abdel from Amsterdam. He's going to tell us a little bit from a different perspective from like genetics, and we're going to include the environment in there. So looking forward to this.
Yes, thank you. So genes influence complex traits through environments that differ between geographic regions, is the title of the talk. So we all know in this room how useful genome-wide association studies have been in linking genetic variants with so-called complex traits. So complex traits are traits that are influenced by many genes with very small effects in the environment. So this method has been around for about 15 years, and over the past 5 years, sample sizes have more than tripled, and so have the number of associated loci. Well, I'm not sure if they have tripled, but they have definitely increased. Here we see a summary of the GWASs on major psychiatric disorders done by different groups from the Psychiatric Genomics Consortium. So on the left, you see the Manhattan plots. It's with all the individual SNPs. The bigger these sample sizes get, the more of these SNPs we locate. And it's quite challenging to make sense of these genetic effects on an individual variant level. It's quite rare that, especially for these kinds of psychiatric or behavioral traits, that we find novel insights about biology from a single variant. I think this highest hit from schizophrenia here is one of the rare exceptions where a clear new biological insight was gained. They learned that it had something to do with a synaptic pruning activity that was especially active during adolescence. But so what I like to do is to take the aggregate, the genome-wide aggregate of these signals, and then do analyses on like the trait level, which can also be quite informative. So here in the middle, you see that the aggregate of the genome-wide signals captures quite a substantial part of the heritability that we expect from twin studies. One of the analyses that you can do with the aggregate is looking at genetic correlations, which we saw very sophisticated approaches in the previous talk as well. And basically, the big conclusion is that a lot of these genetic effects are shared. If you have a higher risk for, let's say, bipolar disorder, you also have a higher genetic risk for schizophrenia. So all the way on the right, I've put some other traits, the social and cognition traits, so intelligence, educational attainment, and income. These are almost all red, because a higher genetic risk for a psychiatric disorder generally is not good for your, for how good you do in school. But there are a couple of interesting exceptions. Autism, bipolar disorder, and anorexia show a positive genetic correlation with educational attainment. We're not exactly sure yet why, so there are some approaches that have tried to shed some light. One of them is by using polygenic scores. So polygenic scores is, as most of you will know, the sum of your risk alleles weighted by the effects as estimated in a GWAS. So we, for example, published a paper maybe 6-7 years ago where we looked at the polygenic scores for bipolar disorder and schizophrenia and whether they predict whether someone has a creative job later or not. And we found that that was the case in the Netherlands, in Norway, and in Iceland. So this is just one of the many ways you can use polygenic scores in a research setting. In the talk later, I will show you some other ways where they have been useful. There's also quite promising, according to some, not according to everyone, for clinical purposes. the bigger these GWASs get, the more predictive these polygenic scores get. And if you could use them in the clinic for stratifying people on genetic risk, that could potentially be useful. And we see that the bigger these GWASs get, the more precise these effect estimates get, and the more predictive these polygenic scores get. So in this paper, we said when the nature of the predictive ability of polygenic scores for behavioral traits or psychiatric traits is not fully understood, applying them in either scientific research or the clinic can lead to incorrect interpretations and conclusions. So as I said, the predictive power of these polygenic scores keep increasing as the GWASs get bigger. But we don't really have a good idea of why these polygenic scores are predictive. We don't, like, there are no genes that make you schizophrenic or that encode for a high school diploma, or they influence very basal and the complex biological processes at the cellular level. These influence all kinds of traits, and those traits influence your environment, and what environment you get exposed to, that environment influences you again, and that also influences even your biology, and all of that gets captured in this GWAS signal. And so in this paper we try to discuss how to go ahead to disentangle all these different sources of GWAS signals. So I think many of you know this movie. Can I see hands who actually saw this movie? Okay, that's actually quite, I think maybe even more than half. So it's a movie from 1997 about a dystopian future society where babies are only allowed to be born if they have been screened for their genetic risk. If they've been screened for polygenic scores, pretty much. The term polygenic score did not exist back then, but the movie is pretty much about polygenic scores. And as many of you probably also know, that the movie it's not entirely science fiction anymore. There are companies now that are offering to screen your embryo at an IVF procedure for all kinds of disease traits. This very cute baby called Aurea Ian Mai Smigrotsky, I think she is maybe 2.5 years old now. This is the first baby that has been born from such a procedure. It says here, Aurea doesn't know it yet, but her birth was very special. She is the world's first PGTP baby, meaning she is statistically less likely than the rest of us to develop a genetic disease or disorder throughout her life. So this is from the frequently asked question section from the website of this company that offers this. It says, does genomic prediction screen purely cosmetic traits? No, we only provide we only provide risk scores for polygenic traits related to diseases, not for purely cosmetic traits such as eye color and eye color. Our goal is to provide improved health to IVF families. It says, does genomic prediction clinical laboratory screen embryos for intelligence or IQ? They say no. So I made this figure using GWASs from the traits that this company uses to screen their embryos. So that's over here. It includes brain disorders, Alzheimer's disease, schizophrenia, heart disease, all types of cancers, and inflammatory bowel disease and diabetes. And then here on the y-axis are all dimensions that are not disease traits. These are just normal dimensions of variation, including height and BMI, reproductive traits like number of children or age at menarche, and then the substance use traits, how many drinks do you have a day, psychological traits like neuroticism, risk-taking, loneliness, educational attainment, and also IQ. So the empty boxes are not significant, and if it's not empty, then there's a significant genetic correlation. So this means that these companies are not, if they are selecting on these diseases, they are not only selecting on diseases. They are also selecting on IQ, and not even always in the same direction. So why are these genetic correlations there? So probably substantial part will be some pleiotropy or traits that are being influenced that have an effect on other traits. But a part of it is also because of environmental influences that are captured in these GWASs, specifically gene-environment correlations. So we have already been aware of these gene-environment correlations on a family level for a while. So in this is a very nice paper from Rosa Cheeseman and Jerome Breen, who is also going to give a talk tomorrow. Here they showed that a polygenic score for educational attainment predicts educational attainment about twice as well in non-adopted people than in adopted people. And that is because these polygenic scores predict on a family level the genetic and the environmental influences. Because if you grow up with your biological parents, you share all of your genetics with them, but if they have a higher polygenic score for educational attainment, they will also have better resources to support you in your environment to do better in school. So in this paper, we started to suspect, or actually already saw, that these gene-environment correlations extend beyond the family. So what we set out to do in this paper is just look at the geographic distribution of polygenic scores. So I made polygenic scores for more than 30 traits. So these are physical and mental health outcomes, things like personality, just height, BMI, substance use. We tried to get as many GWAS summary statistics that did not include UK Biobank, so we could use those to make polygenic scores in UK Biobank. And these are for 450,000 UK Biobank participants with European ancestry. So before controlling for ancestry differences, a lot of them were clustered geographically quite significantly. So this x-axis here is the Moran's I. It's a measure for how geographically random things are. It's a spatial autocorrelation. So 0 means random, 1 means perfectly clustered. And before controlling for ancestry, we saw geographic distributions that looked pretty much like the ancestry differences within the UK. So these are the first 3 PCs. More than 80% of those first 100 PCs show some significant correlation with geography, often separating Wales from England and Scotland. So cultural barriers are often pretty well visible in these PCs. But after controlling for these principal components for the first 100, all of the geographic clustering dropped for pretty much all of the traits except for one outlier, and that was educational attainment. So if we plot educational attainment polygenic score on a map, it looks very similar to the map of socioeconomic differences in the country. These black lines are coal mining regions. The coal mining industry collapsed in the previous century between the 30s and the 80s. A lot of economic problems followed, a lot of joblessness, a lot of all kinds of stressors. And what we saw was that people that were born in those coal mining areas and migrated out, they have a higher polygenic score on average than the entire country. And the people the whole coal mining region on average has the lowest, but if you take out the people that migrate out of there, they have the higher than the rest of the country. So there's sort of a brain drain going on that is detectable on the genetic level. I regret not including the longitudinal version of this figure where you can see that this difference is present throughout the entire century, and you can also see the genetic differences growing between those regions because of that brain drain. And so these regional genetic differences are not random, they are clustering with environmental circumstances. This is the map of clearly an environmental factor, namely the number of fast food outlets in your neighborhood. It shows the same distribution as those polygenic scores, right? So there are big environmental differences. It's very different living in a poor neighborhood than in a rich neighborhood. So then in this paper, we try to tease this apart in a bit more detail. We try to, well, in the first part, we used sibling pairs in UK Biobank to try to figure out whether these gene-environment correlations extend beyond the family. And then in the second part, we try to see whether we can control for these gene-environment correlations in GWAS. So we did both of these analyses, both of these parts in more than 50 complex traits. So we did it in about 250,000 UK Biobank participants of European descent, so it's lower than the 450 because they had to have all of these regional measures and phenotypic measures. And these are the 56 complex traits that we looked at. So I'm not sure if you can read all of them, but they include anthropomorphic traits, things like height, BMI, cardiovascular diseases, cognition and SES, things like educational attainment, income, intelligence, depression measures, other behavioral traits like risk-taking or time spent watching TV, physical health outcomes, reproduction, sleep outcomes, social outcomes like loneliness, and a bunch of substance use outcomes. So what this graph shows, this is no genetic data yet. This is just how much variation is explained by where people live. And all of these are significant. So where people live has an effect, or is associated at least with all of these outcomes, some more than others. And in general, current address more than birthplace, probably because of migration. It's especially visible here with this is household income. So current address explains 15% of the individual differences and a lot more than birthplace, because if you can afford to go live in a better neighborhood, you move there and you are surrounded with a lot of other people in the same situation. So, okay, let's start with the first part. So just to explain conceptually what we try to do here. So here we see a man and a woman. The man managed to convince a woman with a higher polygenic score to come live with him and to have not one but two babies. So they pass down their genetics, but also the environment. And that environment is correlated with their genetics. Because if the parents have a higher polygenic score for educational attainment, they will probably also have better resources to provide a more supportive environment. And that's what we call passive gene-environment correlation, because they are just born in that family. And when they grow up, they move to different regions, and there are small genetic differences between the siblings as well. And the sibling with the higher polygenic score is more likely to move to a neighborhood with more healthy environmental influences, and that is what we call active gene-environment correlations. So we used a bunch of models to try to separate these two. I'm just going to talk about one because I don't have that long. So it's based on this paper where they looked at within and between family polygenic score prediction in young children. So this is a model just for individual level polygenic score prediction. And here they added a between family effect, so just the average polygenic score for the two siblings in the family. And they saw that the predictive power of polygenic score for especially intelligence and educational attainment was 60% greater between families than within families, because between families it also predicts that environment. It predicts both genes and environment. So we did the same thing, but in adult pairs, 26,000 in UK Biobank. So when you add that Oh, sorry, and we asked, are there any additional gene-environment correlations at the regional level that are not captured at this family level? So when you add this between family effect to the model, the within family effect decreases, because if you don't include the between family effect, then the within family effect also includes those environmental effects. And we asked if you then also add the between region effect, does that decrease that within family effect even more, even further? So we did this with the polygenic score for educational attainment and predicted 56, those 56 traits. And we saw for, indeed, that in general, the within family effect did decrease. So here on the x-axis is the within family effect with only the between family effect, and then here on the y-axis we added that region effect. And so this was significant for 10 traits, the traits that have the names here. But the 5 most significant ones were not educational attainment. This is an educational attainment polygenic score. And the reason is probably because these are traits that are more subject to change after the siblings have migrated out. At an adult age, they probably have finished a lot of their education already, but their BMI, for example, can still change. Okay, so for the second part, we did a GWAS on those same 56 complex traits with and without controlling for geography, and then looked at what happened with the signal. So here we see what happened to the SNP-based heritability. The stars indicate that it significantly decreased, and that's only for a relatively small number of traits: educational attainment, intelligence, income, time spent watching TV, overall health, and age at first birth. So these are traits that are quite closely related to socioeconomic status. But if we look at the change in their genetic correlation with educational attainment, then these stars showed almost all of the traits show a decrease in their genetic correlation for educational attainment. So while their heritability doesn't decrease significantly, the content of the signal changes in that you remove that educational attainment signal. Most significantly, this was for traits related to BMI and substance use, so stuff that you put into your body. We saw the same thing happen with household income. Actually, the genetic correlation between educational attainment and income is about 0.9. So they capture almost the same signal. I think it's just these are just proxies for socioeconomic status. Okay, so I think what I think we can conclude is that we as a society, we make genetic effects stronger. We reward certain genetic propensities with a better environment and punish the lack of certain propensities with a worse environment. If we see that the young kid is smart and talented, we send him to a better school where he gets more support to develop his talents. When he grows up, he gets a better job so he can afford better housing in a better neighborhood. And that makes society more unequal than it already is. I mean, what genes you inherit is already a lottery, and then we reward the winner and punish the losers, if you will, so giving them a double advantage and double disadvantage. And this also makes studying genetics more difficult, because it gets more difficult to separate genetic from environmental effects. I'm not even sure, like these, correcting for these regions decreases the genetic correlation, but it doesn't remove it. These geographic regions are just a proxy for your actual social environment. I'm not even sure if we should always control for it. It depends on what you want to do with that signal. If you just want to create a polygenic score and maximize its predictive value within the same social environment, then probably you shouldn't control for it. But if you want to claim that, let's say you select embryos purely on the trait that you say you selected on, then probably you should. So I want to thank the co-authors of at least the last paper. There should be more names on this actually from the other work as well. And I want to thank especially UK Biobank very much for making their data, the great data set public. Without them, we wouldn't be able to do this work. And thank you also for inviting me and for your attention. I was aiming to get it at 0, but
Any questions?
Hi, I really enjoyed your talk. It was really, really interesting. So my question is, since you are looking at that gene-environment interactions, have you considered doing an epigenome-wide association study instead of using GWAS?
No, simply because I don't have that data for this many people. Yeah, I don't know if, yeah, it would be interesting to look at how epigenetics associate with this, especially on, I would love to make geographic maps of epigenetics and see what that looked like. But I simply don't have the data. If someone else has it, then I would be very curious to see what that looks like. Yeah. Yeah.
So I was, Mina, I mean, that was brilliant. That was really fascinating, I have to say. I mean, I think there was just a couple of things there are more comments really, more than anything else. I think one, how important this kind of information is, because I think currently there's a misinterpretation of the importance of some of these genetic factors, because you're absolutely right, the genetics is, the environment is making the genetics appear stronger than it is in its raw sense. But you're worried about policymakers assuming that for educational attainment, you know, polygenic risk scoring is useful or anything of that kind. It was even touted in the UK by Dominic Cummings at one stage that PRS scores for education should be used for stratification of children into schooling. So, I mean, I think making this kind of work, I think, is much more important in terms of ensuring that there's a proper understanding. And I think at the moment, from a clinical perspective, I think there's a lot of patients who are thinking of a kind of more genetic determinant kind of model, and that's being encouraged in some senses. And I think it needs to be counteracted because, of course, some of the things you're talking about in terms of environment are eminently treatable in some sense by changes in government policy. Yeah. And that's really good to disentangle. So, I mean, it was just to say how you know, I think this kind of work needs much more of a platform. And yeah, and I guess if there's been a New England Journal of Medicine paper actually about polygenic risk scoring, you probably know already, and usage of polygenic risk scores for embryo selection, which I have to say in the UK, I'm really glad to say is not legal. Yeah. But yeah.
Yeah. Yeah, those are very good points. It's a lot of what we are picking up says not just something about the traits, but also about how your society is organized. That I would love to see some of this work maybe replicated in countries with a little bit of a different system than the UK, like Norway or, yeah.
Actually, sorry, just one other point. One thing I wondered about, particularly with the housing, was that actually the UK used to have much, much more council housing stock, and that meant that actually you could be living in a relatively wealthy area despite not being wealthy yourself. Over the Thatcherite era, that actually all got, you know, that got privatized in effect. I know I'm sounding a bit political, but still. But, like, but you know, in a way, that's probably a kind of point in time where the selling off of council houses. I wonder whether you could even, I mean, like your analyses, start taking account of those government policy decisions that might have heavily impacted these kinds of factors. You know, public health in effect.
Yeah. Yeah, so we do see like the inequality increasing over time. I'm not sure what.
You probably haven't got enough fine grain to be able to see it.
Or we got an online question first.
Yeah.
And then,
so actually, it's very much related to this one, and it's, so the question is, how is this best translated for governments to improve fairness or conditions for their people?
Sorry, can you repeat that?
How is this best translated for governments to improve fairness or conditions for their peoples? So these findings, how they can be used in a way by the governments?
I don't know. Like I'm not a policy expert. I also try to stay politically as neutral as I can in reporting these findings. they give you a sense of the inequality and the growing inequality, these findings. And I think we need more data from different countries. I already said it, with different, like maybe more egalitarian systems. The UK is one of the most unequal, I think the most unequal country in Northern Europe at least. Yeah, so I hope this stimulates researchers to look at different societies as well.
Yeah, we got one more question, then we need to go.
Yeah, so I mean, Abdel's last, I do recommend like not having a monarchy, right? That probably helps. But I would say that as I'm Irish, right? So, yeah, but Ireland is noticeably more equal than the UK now. But anyway, let's not go there. So, it's two comments. So one is in our online recruitment for depression, we actually see an excess of recruitment, I think, in the coal mining areas,
Okay.
which is interesting.
Yeah.
But also, if you often reflected that we're asking, let's say in schizophrenia, we ask people to read 4 or 5 page consent forms that are written in legal language, and the schizophrenia cases going into our GWASs are mostly,
Yeah.
not always, of course, but mostly
Yeah.
weird.
Yeah. Yeah, I understand what you're saying, yeah. Yeah, so there is also in UK Biobank in general a bias towards higher educated people that participate. We compared some of the things that we find with census data, and we saw that in census data, like, the differences were bigger than that we saw in UK Biobank. Yeah, so we're probably underestimating the effects that I'm reporting because of what you say. Yeah.
Awesome. Thank you very much.